So hello friends, welcome back to my channel Medicine of 101. Today we will talk about resting membrane potential. So it's very easy as far as the topic is concerned from the cell membrane physiology. We are going to talk about this topic, okay? See what is resting membrane potentials and what is action potential. These two terms should be clear, very clear, okay? Before studying the topic. Now, see whenever we talk about the resting membrane potential, that means the potential that we'll see within a cell under the resting conditions. That means whenever we see there is no activity is going on within the cells, the cell is under the resting conditions. At the time, whatever the potential we will get within the cell, that will be known as what resting membrane potential. And whatever the potential, the action potential, that will be seen whenever the cell that will be stimulated from the whenever the cell that will be stimulated for whatever the regions, and whenever the electrochemical changes we will see within that cell after the stimulations, that is known as what action potentials, and the electrochemical changes that will propagate along with the surface of the membrane of the cells, okay, in these things as far as the action potential is concerned. Now see, whenever you are talking in terms of first the resting membrane potential, now the resting membrane potential, this is found in almost all type of the cells. Now the resting membrane potential we can see in all type of all cells, okay. So the resting membrane potential, I write briefly RMP. Resting membrane potential, it is found in almost cell. So almost in all cells we can see this type. Now what about the action potential? Now the action potential, where can we see? The action potential we can see in terms of the muscular cells, so that means within the muscles. Within the muscles where we can specifically talk about skeletal muscles. We can see where in terms of the smooth muscle. We can see in terms of cardiac muscle. Okay, and definitely we will also see the action potential where in terms of the in terms of neurons. So where you will see neurons, and definitely we can see if we just do the combinations, we can write it over here in terms of the neurons as well. Okay. Now I'm going to say in details about the resting membrane potential. So how the resting membrane potential is originated, or how the resting membrane potential is seen, or how it is created within the cells, that I'm going to talk about. Now for that thing, I'll just take a hypothetical cell over here. See, I'm just taking a hypothetical cell, just like this. Okay, let me show over here. Okay. Now see, within the cells there is what nucleus. Definitely nucleus. Within the nucleus there is what genetic material, and there are specific type of the genes that will produce the different types of the proteins. Okay, let me show some genes over here. Okay. Now see, what do we know under normal circumstances? If we just see that the concentration of the different types of the ions within the cell and outside of the cell, they're different. Now, as we know, as far as the evolution is concerned, that the first origin of the life that was found within the sea. So, if we just consider that thing with this thing, that if the originations of the life within the sea, so definitely what around the sea there is what salty water. So, definitely outside the cell, we can consider just like this for understanding purpose. That outside of the cell, that will be what salty. That means outside of the cell, the concentration for the sodium, that will be more. So, outside of the cell, we will see the concentration for the sodium, that will be more. And we know that what we can consider the cell as a bag of potassium. We can consider the cell as the bag of potassium. That means the concentration of the potassium within the cell, that is greater than the outside of the cell. Now, if you just say in this way that the concentration of the potassium over here around 140 millimole per liter, and outside of the cell the concentration for the potassium around 4 millimole per liter, just like this. Okay. Now see, whenever I have already mentioned about here that the genetic material, they produce the different types of the proteins. For example, I am showing over here from this gene what we are getting the mRNA, and from the mRNA we are getting one type of the protein that will just get over here, that will come over here and get fused with the membrane. Now see, for example, the name of the protein over here. Okay. What's the function for this type of the protein? Is the main function of this protein that it will throw out the free sodium outside of the cell, and it will take to potassium within the cell. Now, what did I say you before that? The concentrations of the potassium outside the cell, that is what? More. And the concentration of the what? Sodium within the cell, that is less. That means the cell is throwing out the sodium against the concentration gradient, because the concentration of the sodium within the cell is less. The concentration of the sodium outside of the cell is more. So whenever the cell is throwing out the sodium from inside of the cells to the outside of the cell, that means it is throwing against the concentration gradient. In the same way, see, I have already said you that the concentration of the potassium outside of the cell, that is what, less, okay, and the concentration of the potassium within the cell, that is more. In the same way, we can see that the cell is taking the potassium within them against the concentration gradient. And we know whenever we see any type of the transport within the cell against the concentration gradient at the time, that should be what, the active process. And whenever you are talking in terms of the active process, there should be what, the energy utilizations. The energy in the definitely here will also what, energy utilized. So what of the energy, now see here we will get the ATP that will convert into ATP plus phosphate. Okay, that means this type of the what, energy that will be utilized. Now see, whenever we are seeing this type of the thing, the sodium is going out and the potassium is taken within the cell, like this will get multiple, okay, this type of the pump like the sodium potassium ATPase, where we will also see that the sodium that will be thrown out and the potassium that will be taken within the cell. Okay, now see, just like this, whenever we are just seeing, if we just consider if the pump works for 1,000 times, for example, I am saying, then the 3,000 molecules of the cation that is going out and the 2,000 of the molecule that is going in. As a result, we can see the deficiency of the cation, cations were within the cell. As a result, we will see what, some negative, so we will get some negative voltage over here. And for these things, I am just assuming over here that I am getting around minus 5 millivolt. So whenever the sodium potassium ATPase pump is working, as a result, it is throwing the sodium outside of the cell. It is taking potassium within the cell. As a result, we are seeing there is deficiency of the cation within the cells. I am just considering it around minus 5 millivolt for these things. Okay. Now see, whenever we will see the concentration, whenever the potassium that will be taken within the cell, as a result, we will see the concentration of the potassium that will keep on increasing. The concentration of the potassium that will keep on increasing over here. Now, whenever the potassium will increase over here, now see, with respect to the inside of the cells, with respect to the outside of the cell, with respect to potassium over here, if we say, then the concentration gradient, the potassium is supposed to go from inside of the cells to the outside of the cells. For that thing, we will see within the cell, there one molecule that will get activated over here, and that will produce the mRNA, and that will produce another type of the protein, another type of the channel. Okay, that is known as ungated. Okay, there is known as ungated potassium channel. As far as the channel is concerned, let me say in details about the channel over here. Now, whenever we are just talking in terms of the channel, see, if we just talk a little bit of them, so we will get the different types of the channel. First, we can get the ungated channel, or another name for it is known as leaky channel. Okay, they are freely permeable. Now another type of the channel we will get, for example, is the leaky channel. Another type of the channel we will get, okay, just like this, and these are voltage sensitive ion channel. That means this channel will get open and closed during the voltage sensation. If there is any changes in the voltage, then this channel will open, and if there is change in the voltage sensation, we will see this channel will get closed. So this is known as what? Voltage sensitive ion channel. Voltage sensitive ion channel. Another type of the channel we will get over here. Okay, where we will get like this. This channel will get opened whenever any type of the substance, for example, any type of the substance, it may be the hormone, it may be the chemical, the different types of the secondary messenger that will come over here and that will fuse, then this channel will open. So it is known as what? Ligand channel. You can see it is known as ligand channel. It is known as ligand channel. Now see, as far as the what as we are talking over here, the ungated channel, that means this leaky channel. As a result, whenever the channel will be leaky, in these cases we will see there will be efflux of the potassium. Okay, just like this. Let me show one more over here. Okay, now see, just like this, there will see, okay, many within the cells. There will get many what ungated potassium channel. That means what the voltage, that's the leaky potassium channel over here. Okay. For example, now we'll see in these cases what the potassium that will diffuse outside of the cell. That will diffuse outside of the cell. That means the potassium that will come from what inside of the cells to the outside of the cell. As a result, we will see this voltage. Okay, what the cell within the cell we will see there will be more negative. That means from minus 5 millivolt it will go to minus 20 millivolt. For example, minus 10 millivolt first, minus 20 millivolt, minus 30 millivolt, minus 40 millivolt, just like this. Why the potassium is going out? Because of the concentration gradient. Because the concentrations of the potassium within the cell is much more greater than the concentration of the potassium outside of the cell. As a result, we will see there will be efflux of the potassium in these cases. Okay. Now see, whenever the potassium is going out, at that time we will see that whatever the electric potential over here, the electric gradient, that means the negative. Potassium is what? It's the positive cation, that means the cation, and whenever we are getting the what negative voltage over here, this electric potential that will make the potassium to go outside. But in the initial conditions, we will see the concentration gradient. If we just consider the concentration gradient in this way, let me show over here. The concentration gradient of the potassium, and for example, I am showing this one with the electrical gradient for the negative, that will not let the potassium to go out. See, in these cases we will see in the initial stage, because only for minus 5 millivolt. In the initial state, what we will see over here, the concentration gradient that will be greater than the electrical gradient. As a result, we will see the potassium efflux. We will see the potassium efflux. Okay. Now just like this way, okay, the potassium that will diffuse. That means what? That is known as the diffuse potential of the potassium. That is known as what? Diffuse potential of the potassium. So we will get it over here. The diffuse potential of potassium. As a result, there will be what? Efflux of the potassium. Now just like this, it will come around whenever it is around minus 40, minus 50 millivolt, minus 60. Okay. Now at a time we will see the what we'll see the negative polarity. That means the voltage, negative voltage, that will get increasing where within the inside of the cells. At a time we'll get whenever we'll see the concentration gradient. The concentration gradient that will be equivalent to that will be equals to the electrical gradient. And whenever we'll get the concentration gradient that equals to the electrical gradient of the potassium in these cases, there will be no net movement of the potassium. That means there will be no movement of the potassium from inside of the cells to the outside of the cell. So in these cases, there will be no net movement of potassium. No net movement of the potassium ion. Now in these cases, see, for example, in these conditions, we got over here. Let me show. Whatever the electric concentration gradient over here, in the same way, we are getting the what? Electrical gradient. In this case, if we see there is a concentration gradient that is equal to the electrical gradient, now there will be no movement of the what? Potassium. And in these conditions, we will get around minus 85 millivolt. Minus 85 millivolt will get just like this. Now these potentials, the potential at which we will see the concentration gradient that will equals to the electrical gradient, that is known as Nernst potential or equilibrium potential for the potassium. Now let me show over here this thing. Whenever we will get the concentration gradient that is equals to electrical gradient, okay, this potential that will be known as equilibrium potential. Equilibrium potential, or another name because of the scientist name, Nernst potential. Nernst potential. So we will talk about the equations in the subsequent part. Okay. So at this potential, we will see there will be no net movement of the what? Potassium from inside of the cells to the outside of the cells. In these conditions, we will get around minus 85 millivolt. Now we can say for the efflux of the potassium in these cases, we will get the resting membrane potentials of the cells around minus 85 millivolt. So Resting membrane potentials will get in these cases from, it's around 85 millivolt to 90 millivolt. Okay. So we'll get the resting membrane potentials. Just like this, we'll get there is genesis of the what resting membrane potential within the cell. And what about sodium? See, whenever the sodium is going out, so the concentration of the sodium outside of the cell, that will be more. As far as the concentration gradient is concerned, so as a result we know that if the concentration of the sodium outside of the cell is more, so there is a chances for the sodium to go back within the cell again. But this channel that they will not allow the sodium to go within the cell. So as a result that there is no chances for the sodium to go within the cell. And by these processes we will just do what we will see the resting membrane potential genesis. Now we'll talk about another type of the cells. Let me show over here. For example, there is another type of the cell. Within the cells, again the nuclear material, genetic materials are over here. They will produce the different types of the protein. Okay, and already I have explained these things that by the action of what sodium potassium ATPase, okay, the 3 sodium that will be thrown out of the cell and 2 potassium that will be taken within the cell. 3 sodium that will be thrown outside of the cell and 2 potassium that will be taken within the cell. Now see these things, okay. Whenever the sodium is coming out, as a result the concentration of the sodium outside of the cell that will be more. That will be more over here, okay. Now around for this we will get how much? Around minus 5 millivolt. First we got, okay. Then we got around what? The resting membrane potentials we got around minus 90 millivolt, for example. Okay. Now in this case, whatever the thing we'll see. For example, there is one gene. These genes will produce a mRNA, and this mRNA, okay, will produce definitely a protein, transmembrane protein over here. And for example, they are producing a protein, and this protein is coming over here. And in this case, whatever thing we are getting over here, see, for example, this is a sodium channel. Now, whenever the sodium channel is opened over here, we'll see that efflux of the, sodium, within the cells. There will be efflux of the sodium in this case, okay. Now see why in these cases we are seeing the concentration gradient. See the concentration gradient. First we will get the concentration gradient and also the electrical gradient. Why? These both things will work over here. Now see why I am saying this thing. Because whenever the sodium is outside of the cell, at that time if we just see what type of the thing we are seeing within the cell there is what? Negative polarization. That means the potential there is what? Negative. And the concentration of the sodium is outside of the cell, what is the potential is positive, or inside of the cell the potential is negative. And that the other thing is also over here that the concentration of the sodium outside of the cell is more, the concentration of the sodium within the cell is less. So if we see the two things that will work over here, the concentration gradient plus the what? Electrical gradient. That means the both things will work over here. As a result, we will see the influx of sodium. And whenever the influx of the sodium we will see over here, now these things from minus 90 millivolt, minus 40 millivolt, minus 30 millivolt, minus 20, just like this it will come around 0. Then after that, for example, we will get over here plus 10 millivolt, plus 20 millivolt, plus 30 millivolt, just like this, plus 40 millivolt, and around at last we will get around 65 millivolt. Now see, I will talk about all these things, how we are getting 65 millivolt. Whenever the sodium is coming outside of the cells to inside of the cell, as a result, it will take the resting membrane potentials from minus 90 millivolt to 0, because it will neutralize the negative polarizations. Okay, it will just neutralize the negative polarizations. That will we will see what type of thing we will see over here. We will see the depolarizations. We will see whenever the sodium that will influx within the cell. As a result, we will see the depolarization of the cell. Now see, whenever the sodium we are getting within the cell in these cases, see. Outside of the cell is still the concentration of the what? Sodium is more, so concentration gradient is more, and we will see right now for example 10 millivolt. Now we will see in these cases there will be repulsion, because why we will see this type of the repulsion. See this, try to understand from here. This is positive, right? Plus 10 millivolt, and this is also what? It is the cation. Now whenever we are seeing the cation over here, so it will just do the repulsions, because it will not let the cation to go again. within the cell, because already the cation is over here. Now in these cases we will see the concentration gradient. Okay, a time will come where we will see the concentration gradient. Okay, the concentration gradient will be equals to the electrical gradient with respect to the sodium. Now see, after that, whenever we will get the concentration gradient outside of the cell for the sodium, that will be equals to the electrical gradient of the sodium within the cell. As a result, we will see there will be no net movement of the sodium. So here we will see no net movement of sodium. Now whenever we are seeing there is no net movement of the sodium in these cases, so that is known as what? The equilibrium potential for sodium. That is known as equilibrium potential for sodium, or we can say what? We can say the Nernst potential. You can write it over here, equilibrium potential, or you can say another term for the scientist, Nernst potential, and we will get around plus 65 millivolt. Whenever it will be just closer to plus 65 millivolt, at the time we will see there will be closure of this channel. Whenever it will be tends to reach around plus 65 millivolt, at the time we will see the closure of the sodium channel. So there will be no further influx of the sodium within the cell. Okay. Now see, whenever the sodium is going for the concentration gradient and also the what? Electrical gradient, we will see what there is a diffusion of the sodium. There is diffusion of sodium from outside of the cells to inside of the cells. This is known as what? Also the diffusion potential for the sodium. Okay, so that's all for today in terms of the resting membrane potentials. We will see this type of thing, especially in terms of the action potentials. We will talk about these things in the subsequent parts of the videos. Now today just focused over here that how we are getting the genesis of resting membrane potential, because of the what? In a nutshell, if you want to say, that we will get the efflux of the potassium. And whenever we will get the efflux of the potassium, as a result we will see there is genesis of the resting membrane potential. Okay, so see you in the next video very soon. And if you think this video is helpful for you, please do like and subscribe and try to share with among your friends. We'll see you in the next video.